Okay, hi everybody. I am at NAU. This is where I teach at NAU. This is our Bio 182 lab. But I wanted to take a minute to get us prepped for next week. So we have a lot of stuff to cover, and I wanted to spend more of our time doing some hands-on experiments. And so I thought I would do some of this lecture for this section about plasma membranes, do it as a video so that would free us up some time in class to do some experiments. So let me share my screen with you guys. And okay, put this on the slideshow. So we're going to switch over to chapter 5. So we're not skipping chapter 4. We'll come back to it. But we're going to focus on plasma membranes. And this first section is about its components and its structure. And so this is an active link, and we'll take you to that section in the textbook. Okay, so let's talk about plasma membranes. The things that you guys are going to want to know, and will be on our next exam, is to understand the cell membrane fluid mosaic model. So I'll tell you what that is. Describe how phospholipids, proteins, and carbohydrates work together to maintain the fluidity of the plasma membrane. And then why is fluidity of the membrane so important? Okay. And so remember back on the very first day of class, we talked about how biology is really split into various levels of outside, inside, and interface. And so what we're talking about now is the interface of that epicellular level, right? So here I've got on this image is of different plant cells. And so if you can see my mouse cursor, here is one of the plant cells. This is the inside of the plant cell. These little kind of triangle-y spaces or even square spaces in between, that would represent the outside of the cell environment. And then of course, what this is appearing as the black line is the interface. That's the plasma membrane that's separating that outside of the cell from the inside of the cell. And this interface at the cellular level is so important for maintenance of cellular function because it controls what's coming in and out of the cell. It gives the cell its shape and its structure, and it has to be able to be flexible and pliable. And so like our red blood cells have to be able to squeeze down to be able to fit through our capillaries because that's such a small space. So that plasma membrane has to be able to be flexible and pliable enough so that it can change its shape, so that it can perform its function. Now, the other thing that the plasma membrane does is it carries markers so that it can recognize itself. It can recognize that, oh, this is another cell of the same individual that, so this cell is on my team, and so I don't need to attack it. And then it also needs to be able to say, okay, this is a foreign entity, and I need to do something to get rid of this intruder. And so I think we're all pretty familiar by now with the example of viruses attaching to and then taking over our cellular structure in order for it to reproduce. For the virus to reproduce, it has to commandeer our cellular functions because it can't do it on its own. So our cells, that's what our immune response is our cells recognizing that this virus, let's say the coronavirus, doesn't belong here. And so we can, we know that we need to do something about it. So, and we'll talk about exactly kind of how that works in a bit. So back when, in the early 1900s, I think 1915, oh yeah, 1915, The structure of the plasma membrane was discovered, and at the time they thought it was kind of two layers of proteins with this a layer of lipids in the middle. And so they used this analogy of the sandwich where the proteins were the bread, and then the lipids were whatever the filling was, in this case, the yummy, yummy grilled cheese. Look at how melty that is and delicious. I think I might be hungry. But as technology advanced, we got a better understanding of the cellular components. And so this is the importance of science and not being afraid of being wrong, and being able to say, okay, based on this new evidence, we're going to modify what we think this plasma membrane is. So as technology advanced, we started to understand it's more of a mosaic that makes up the cellular membrane. There's a lot of components at play here. And just like each of these individual tiles gives us, it's those individual tiles are able to maintain their identity, but they all contribute to this overall image of this cat overlooking the sunset out on the ocean. Okay. So now we use, instead of using the sandwich analogy, we say that we're going to talk about the fluid mosaic model. And this is telling us that the plasma membrane is a mosaic of a lot of different components. These are phospholipids. Remember how cool phospholipids are. These are my favorite biomolecules because they have both a hydrophilic end on that phosphate head, and then they have a hydrophobic tail, and then that gives it, having both hydrophilic and hydrophobic properties, gives it a unique way that it interacts with the environment. Okay, so that's one of the major components. Another one are sterols. In animals, we use cholesterol, fungi use ergosterol, and plants use, I think it's called phytosol or something like that. So, but they all these sterols serve the same function. We also have proteins that are embedded in the plasma membrane, in this phospholipid bilayer. And some of these proteins go all the way through, and some of them just kind of sit at the surface. There's some proteins that have some carbohydrates attached to them. There's some lipids that have carbohydrates attached to them. And so that's our fourth. Carbohydrates is our fourth component of our plasma membranes. So phospholipids and cholesterol, those are both lipids. And then we've got proteins, and then we've got carbohydrates. So in the typical human cell, whatever typical means, our plasma membranes are composed of about half of the components are proteins. 40% are lipids, so that's phospholipids and cholesterol combined. And then the remaining 10% is carbohydrates. So, but this is the typical cell. And so like there's a lot of variation. So depending on the actual job of the cell, there's going to be a different composition. So I think in the book they talked about how your certain types of brain cells have a lot more of one component or another, a lot more proteins and a lot more cholesterol and stuff like that. So know that there's a lot of variation in how plasma membranes are put together depending on what the job of the cell is. So here's a couple of terms that will hopefully look familiar, but we haven't really discussed in class. So glyco, remember glyco, whenever you hear glyco, you think sugar. So glycoproteins, these are molecules that consist of carbohydrates that are attached to proteins. And so here we can see this. And so here's some alpha coils of our secondary structure of proteins. Remember that. Here's a beta sheet, our secondary structure of proteins. And attached to that protein are these different color shapes are representing different types of sugar. But So this is saying that there's a sugar molecule, a bunch of sugar molecules attached to a protein. And so therefore we call it glycoprotein. Similarly, we have a fatty acid here. And then again, these different color shapes are representing different sugars. So this is a glycolipid. It's just a mashup of those two words. So carbohydrates exist only on the surface of the plasma membrane. And remember that sugars are a hydrophilic substance. They love water. And so they're not going to be found in that fatty acid tail section because they don't want to be there. They want to be on the top interacting with the water. All right, so let's go through these components. Phospholipids, remember these guys have a split personality. So this is a word that I did not introduce to you guys originally. I think there was a lot of terminology that people were getting confused about. So phospholipids are amphiphilic. So if you think of amphibians, they have two lives, right? They have a life in the water and they have a life on land. So the way that I like to think of these is that these are amphiphilic. So they have two lives. So they have a hydrophilic head where that phosphate group is. Remember these, this negative charge on this oxygen is going to make it want to interact with the positively charged portions of water molecules. Okay, so there's one of its lives being hydrophilic, and then the other life is being hydrophobic. These fatty acid tails are repelled by water. And so you can see it drawn in different ways here. Okay. So these form a lipid bilayer sphere. And so in class, I used the paper analogy. So you can see here, when we get a collection of these phospholipids, the phospholipid heads interact with each other and the watery environment that's out here. And then the hydrophobic tails interact with each other to form kind of this layer that's being protected by the water from the outside and also the inside of the cell. And so you can see how these phospholipid heads are creating an interior portion that is also filled with a watery substance. Okay, so this gives us our outside, our inside, and our interface. Okay, so now we can switch to the second component, proteins. And again, proteins are the dudes that get stuff done, right? These are the workhorses of the cell. So when we're referring to plasma membranes, we have what we call integral proteins. And this is just referring to the fact that they're completely embedded in the phospholipid bilayer. And so here in our example or diagram, we can see two of these proteins that are extending beyond the phospholipid portion and into the watery environment. This portion of this protein is here embedded in the fatty acid tails of our phospholipids. And then we have a portion of that protein that is sticking out. And so this is completely going through the phospholipid bilayer. Okay. So that's why we call them integral proteins or integens, because they're completely integrated into that membrane structure. So this portion of the protein then has to be hydrophobic in order for it to interact with these hydrophobic fatty acid tails. And then this portion of the protein that's sticking out has to be hydrophilic. And this portion that's sticking out the bottom has to be hydrophilic. So that means that these integral proteins are also amphiphilic. They have a portion that's hydrophilic on either side, and then in the middle, they have a portion of them that is hydrophobic. Okay. And so here we can see just kind of a simplified version of this diagram that's saying, so this little yellow box is supposed to be indicating the fatty acid tails. And we see that, you know, these proteins can come in different shapes and sizes, but we've got a portion of that protein that is hydrophilic, a portion that is hydrophobic, and so on. And they can be very small, or they can be very large, or they can be somewhere in between. Okay, and then the other kind of protein that we see on the associated with the plasma membrane is that we see these peripheral proteins. So these are proteins that are just attached to the surface of the plasma membrane. They don't go into the fatty acid tail portion. So these are only hydrophilic, right? So they can attach to integral proteins, or they can attach to the phospholipids. And the function of these peripheral proteins varies depending on the job of the cell and the job of the protein. And so we call them cell specific, right? Because what they're doing is very specific to what function needs to occur. So they might be enzymes, remember enzymes. They might be providing structure for the, so I think in this case, so these are these little long stringy things are representing the cytoskeleton of the cell. And so we often see that these attach to peripheral proteins as like good anchor points. And so that allows the cell to maintain its shape if you don't have a cell wall like us weirdo animals don't. And they also might allow for recognition of other cells and foreign objects. So here we see a protein here. It's got some sugars on it, so we call it a glycoprotein, and then that is used in recognition. Saying, is this part of me? And if it is, then I can be cool, and if it's not, then I'm gonna be mad about that. Okay. So then we have the other component is, the last component is carbohydrates. So these are sugar-coated cells. So carbohydrates are always on the exterior of the cell. And so they're either hanging out with a protein, and so then we give them the name of glycoprotein, or they're hanging out with a lipid, so then we give them the name of glycolipid. And what the carbohydrates do is it allows communication between the cells and allows them to recognize each other or recognize invaders. So cells are sugar-coated. And when we talk about all the glycolipids and all the glycoproteins on a cell surface, we refer to it as a glycocalyx, which is just fancy science talk that means sugar-coated. As a botanist, this calyx comes up all the time. So this is one of those examples of these words that if you break them into their component parts, then it's easy to recognize and kind of understand what they mean, even if you don't really know exactly. So glyco, we know, means sugar. Calyx means coated. And so then boom, sugar coated. So if you guys haven't already, I would highly encourage you to have like a Word document or an Excel document where you're keeping track of these root words, because that is not only going to make this class a lot easier, but all the classes in biology that you go on to take. If you have this kind of dictionary of these root words, I find that it was vital for my success as an undergrad to be able to recognize the prefixes and the suffixes. Okay, now I'm off my soapbox. Okay, so why do we have sugar-coated cells? It increased for contact with water, because remember, sugars are hydrophilic, and so the water molecules are actually attracted to the water. or the water molecules are attracted to the sugars, which means that there's going to be better absorption of dissolved nutrients in that water. Like, and then also, again, broken record here, allows for recognition of self and non-self. Okay, so all of this is leading up to this really important thing of membrane fluidity. So how fluid is your plasma membrane? And remember that it's this mosaicness of the cell that allows the plasma membrane to be nice and fluid. And if it doesn't have that fluidity, it will be too rigid and it will break. And then all the cellular contents will go out into and then we won't have that outside inside interface anymore. And then the cells can't function. And so then you die, basically. So, so it's important to maintain that fluidity in order to keep the function of the cells. Remember, we talked about how blood cells need to be able to swoosh down in order to get through our capillaries. We need to be able to have fluidity to keep substances moving in and out of the cell and all that stuff. Because if our cells are too rigid, they're going to do this. But we want them to be able to be fluid so that in this case of this balloon, the balloon isn't stressed and stretched and so much that they can actually have insert this needle and have the balloon self-repair. And so when it's punctured by something that's so fine, it actually can reseal itself. And that's really important for membrane function. And so here is a YouTube video showing this for realsies. This is a artificial insemination, but you guys can see here's a needle puncturing the plasma membrane. And so they can insert the sperm into this egg cell. And when they withdraw the needle, it's still the egg cell's plasma membrane is able to seal up, and then it's just fine. It's not actually damaging the cell. And so that's what we want in order to do to maintain that fluidity. Okay, so factors that maintain or that control how fluid it is. This is what stuff is in there, how much proteins there are, how many phospholipids there are, how much sugar there is. Do you have in those fatty acid tails on your plasma membrane, do you have saturated fat or unsaturated fat? And if you don't remember what that is, you need to go back and look it up. The length of those fatty acid chains, if you have short chains, you're actually more fluid than if you have these big long chains. Interesting. And then of course, the temperature of the environment. So remember, we talked about how unsaturated fats can pack together to form like solid butters that we call fats. And then unsaturated fats, because they have that double bond between the carbons, it creates that kink that prevents those fatty acid tails from stacking up too densely. And so if it's cold enough, then we might actually see enough of that stacking up that we might have this membrane rigidity. And the reason that we might see more cholesterol is that cholesterol helps maintain that fluidity even under cold environments. Okay. And so we want to make sure that our plasma membranes don't freeze. And so varying the amount. And so, but then also it could be too fluid, and then it would kind of break apart and not actually provide the structure that is needed. So we want to be able to hit that sweet spot. And depending on what type of environment we've evolved in order to fit with, that might determine how much cholesterol do we need? Do we need saturated or unsaturated fats? How long are our fatty acid chains and all that stuff? Okay. Now, here's a nice little summary table that I yoinked from your book. This is for your ease of studying so that you can think about these different components of the plasma membrane and then know what they do and where they're located. Okay, that concludes this portion of the blah blah. So make sure and watch this, and I will see you guys on Tuesday.